they have to replicate their DNA, then they do their transcription, and the transcription is going from DNA to RNA, and then they do a translation that is from RNA to protein, and then the protein will do its work. So this is what is a dogma, central dogma. So the genetic information found in the DNA is copied and transmitted to cell daughters through DNA replication. So if you have a cell and is replicating, so DNA will be replicated as well. Then the transcription, as I mentioned, is how the RNA is synthesized, and the translation is how the proteins are translated. So let's look at the structure of DNA. We have already seen the building blocks of DNA, which are the deoxynucleotide that we mentioned before. So a DNA is basically a polymer of a deoxyribonucleoside monophosphate we talked last time about. So they are a polymer. It means they are like protein, are formed by amino acids, like carbohydrate are built on the glucose, and the DNA is a polymer of deoxyribonucleosides, and the RNA is a polymer of ribonucleosides. We talked about this last time. So the DNA is a polymer of a deoxyribonucleoside monophosphate, covalently linked. It means it's covalently linked by 3' 5' phosphodiester bond. We've already mentioned this last time, but let's quickly show you the phosphodiester bond. This is the square that you can see here is blue. This is the 3', and we talked about why we call it 3' and 5' in the previous lecture. So this is phospho because there is a at 3' and the 5' there is a phosphate that is linked to bond. That's why we call it di. Di, it means two. So there is 3' here, 5' here from the other sugar, 3' for the sugar on the top, 5' on the sugar below, and these are linked by a phosphate. So phospho is a diester bond. So it's a polymer because it has many nucleotides, 1 nucleotide, 2 nucleotide, 3 nucleotide, etc. So the DNA is a polymer of deoxyribonucleoside monophosphate, covalently linked by 3', 5' phosphodiester bond. It's a double-stranded, you see, when you see DS, it's a double-stranded molecule where there is a two-strand wind around each other, forming a double helix. This is what it means. You have two strands. This is one strand. This is the second strand. They are wind around each other, and it's like a twisted ladder. You see, it's twisted. Okay? In eukaryotic cells, this DNA is associated or bound with different proteins, and that's why we call it nucleoprotein. Nucleo, it means in the nucleus, means the chromosome, and protein. So it's found in the nucleus, of course, and in prokaryotic cells, the DNA is linked to what we call a protein DNA complex in non-membrane bound region. As I mentioned before, we said that in human eukaryotes, we have a nucleus. In bacteria, for instance, we don't have a nucleus. We don't call it a nucleus. We call it a nucleoide, which is a just DNA without nuclear membrane. So what's about the 3', 5' phosphodiester bound? Basically is the bound between the 3' hydroxyl group of deoxypentose of one of the deoxy and the other 5' hydroxyl group of the adjacent. Adjacent, it means next to it. So This is the 3 primes OH, and this is the 5 prime OH of the ribose. One ribose has an OH here, and this ribose at the 5 primes has an OH, and there is a link. So it's a link between the 3 primes hydroxyl, so that it's both hydroxyl, but the difference is just the position. The first one is the 3 primes, and the second pentose is at the 5 primes. So that's why you will have many. You see, this is, as I mentioned, one deoxyribonucleotide linked to another one, linked another one, and they form a polymer of deoxyribonucleotide linked by diester, phosphodiester bonds at the 3 primes, 5 primes. Now, there is one term it's important to know, what we call a backbone. So the base is located along the resulting deoxyribose phosphate backbone are always written in sequence from 5 end to 3 end. So if you hear a backbone, the backbone, it means the phosphate plus the sugar. You see in yellow here. This yellow box is the backbone. The backbone is phosphate and sugar. And as you see here, these are the bases. This is only one strand. And we said there is a double helix. So there is another strand here. So you see here the 5 prime end is a phosphate, and the 3 primes end is a hydroxyl radical. So it's very important, very, very important to know that the direction always from 5 primes to 3 primes. And automatically, you know, at the 5 primes is a phosphate, at the 3 primes is a hydroxyl. Now, the bases located along the resulting deoxyribophosphate backbone are always written in sequence 5 primes end of the chain to the 3 primes end. If you look at this, this is the base, thymine, this is the adenine, this is the cytosine, and guanine. If you want to write it, forget about B and C. We are focusing only about A and D. C, T for thymine, A for adenine, C for cytosine, and G for guanine. We put them together and we write 5 primes. These 5 primes will be written at the T. See here T, and this is a 5 prime, and this is the G, guanine, and this is the 3 prime. So here at the 5 prime is a phosphate, at the 3 primes is OH. So can this phosphodiester bond be cleaved? Yes, it can. So what cleaves this phosphodiester bond? What cut it? Cleaves, it means to cut it. There are what we call nucleases. Deoxyribonucleases are enzymes that can cut the phosphodiester bond in the DNA, and the ribonucleases, they cut the nucleotide in the RNA. Again, this is the double helix that we talked about. So the 2 chain, this is a chain, or you, if you want to call it a, yeah, it's a chain, or I forget the name, other names. Anyway, so these are one chain, 5 primes, 3 primes. The second chain is 3 primes, 5 primes, and these are coiled around. It means twisted around, turned around. All these are the same terminology. So they are twisted around, and they are what we call anti-parallel manner. Why anti-parallel? It means one, it goes 5 primes, 3 primes, and the other chain is 5 primes, 3 primes. So if you look here, the 5 primes on one strand, it has a 3 primes in the other strand. And if you look at the bottom, 3 primes here, and 5 primes here. That's why we call it anti-parallel. They go opposite to each other. Again, you can see the bases are inside C, G, T, A, T, and the backbone, we said the sugar and phosphate are at the outside. So this deoxyribophosphate backbone. The backbone is the sugar and phosphate. And as you can see, when this double helix or the chain are coiled around, there is what we call a minor groove and the major groove. These are just opening in the two chains. When they at the location when they turn around, they make grooves, and one is small and the other is big. So the
دكتور
overall, sorry.
دكتور should the guanine the cytosine should always face the guanine.
Yes, I mean, I'll be talking. It's a very good question. This comes in the next few slides. Very good question. Yes, they will always be together, and I'll talk about it. Thank you. Very good question. So if you look here, what we call spatial relationship between the two strands, basically spatial relationship is just in the three-dimensional space. If you look at DNA, you will find this minor groove and major groove. What you need to know, just there is minor and major grooves, and these are very important because in these grooves, there are some proteins, what we call regulatory proteins, that comes and stick to these grooves, and it helps the gene expression. Again, it can help the gene to be switched on or off. And some of the cancer drugs, like what we call dactinomycin, also known as actinomycin D, it will stick to this grooves and does not allow the gene expression, and therefore the DNA cannot be replicated and the cell will die. So this is a treatment in the cancer. Basically, the dactinomycin stick to the narrow groove, which is a minor. Base pairing, as you just mentioned, you see you have the basis of one strand. This is one strand, the deoxyribose phosphate backbone. And this is the second strand, what we call the anti-parallel. As you can see, 5 here, prime, 5 primes. The other strand is 3 primes, 3 primes, 5 primes. So as you can see, always adenine face thymine, and guanine face cytosine. And these are attached to each other by hydrogen bond. And as you can see, A and T has 2 hydrogen bonds, and G and C has 3 bonds. So if I give you the sequence of one strand, you should get the sequence from the other one. If I give you, let's say, this is C on the first strand, automatically you know the other should be a G. If I give you a T on one strand, the other, it will be A. The bases, as I mentioned, they are linked together by hydrogen bonds, and 2 between A and T. This is thymine and adenine. As you can see, there are 2 hydrogen bonds. And for cytosine and guanine, you have 3 hydrogen bonds. So obviously and clearly, the cytosine guanine bonds are stronger than the thymine adenine. And this will lead us to the how, if we want to separate the DNA strand, we need to cut the bonds, these bonds between A and T. So of course, AT, because they have only 2 hydrogen bonds, it's easy to cut. And the CG bonds are more difficult because they have 3 hydrogen bonds. So can we cut these bonds? Yes, of course we can, using disturbing the pH of a DNA solution. For instance, if we use acidic solution, this will cut these bonds. We can cut the bond as well using heat. High temperature will cut the heat. But importantly, you cannot cut the phosphodiester bonds. So the phosphodiester bonds are not broken by such treatment. And there is one concept you need to know, which is the DNA. When you heat DNA, for instance, if half of the DNA is opened, you call it melting temperature. If someone asks you what is a melting temperature, just you tell is the temperature that allow half of the helical structure of a DNA open. And when the DNA lose its helical structure, we call it denaturation. So is a denaturation like protein denaturation? Denaturation is just when the DNA loses its helical structure, and the melting temperature when is the temperature where half of the helical structure is opened. If you look here at this graph, you can see here at 62 degrees, the melting temperature of a strand 80 will, half of it will open at 62 because it has 80. The DNA that has more GC, half of it require a temperature at 86 degrees. This graph shows how the DNA with GC content needs more temperature than the one with 80. Okay, let's talk about what we call circular DNA molecules. Again, in eukaryotes like human, the human have 2 types of DNA, as we mentioned at the beginning. The one in the nucleus is linear, and you call it double-stranded DNA, and we said is a complex of a DNA and protein. The name of these proteins called histone. When you have your chromosome with the histones is called chromatin. And as we mentioned before, we have in the mitochondria a circular DNA in eukaryotes. In prokaryote, the double-stranded DNA is circular. As you can see here, the eukaryote is linear. In bacteria is circular. And again, in the eukaryotes, they have a circular chromosome as well called plasmid. We've already mentioned this before. We are just talking about now the shape. So in prokaryote, like bacteria, their chromosome is circular. However, in eukaryotes, for instance, they have linear and circular. There is another, the plasmids. What is a plasmid? Basically is a circular chromosome and carry antibiotic resistant genes that can be transferred from one bacteria to another. So some bacteria have DNA that can jump from one bacteria to another, and these DNA called plasmid can carry antibiotic resistant genes. It means genes that if you give someone an antibiotic, it will not be efficient because they have these plasmids. So remember, these are all introduction. We want to see how the DNA is replicating. So we know now that we have 2 strands of DNA in the double helix. And when this DNA, during a cell replication, as you know, one cell will divide into 2 cells. So you have one double strand DNA, and you want this double strand be replicated in that the 2 cells that you will have as well they have 2 chromosomes, 2 double-stranded chromosomes. This process, when you have a replication of the DNA, is called a semi-conservative replication. What does it mean, semi? Semi, it means half. Conservative, you know what is conservative. So basically, this picture will clarify this concept. If you look at this red, or if you want a pink strand, one of them, if you look at one of them, for instance, what we call parental double helix, this is a parental double helix. If you take one strand, and as you can see here, it duplicates into, it gives another gray strand. This is the newly synthesized strand. The other strand as well gives you a newly synthesized strand. So basically, one single parent strand gives another new synthesized. So that's why it's called semi-synthesized, because the cell, will be in a cell. This is one cell, and this is one cell. So the cells, they will have one strand from the parent and one new one. That's why it's called semi-synthesized. So one parental strand is conserved in each of the two new double helices. So when this strand is duplicating, what is the enzymes? What are the enzymes responsible for this synthesis? They are called polymerases. And these polymerases are what we call extraordinary fidelity. What is fidelity? It means when they copy, they will very rarely make a mistake. They are very accurate. That's why we call very high extraordinary fidelity. And we are going to see how this replication occurs in a bacterium called E. coli. And basically, the process of replication is more or less much similar in human, with some differences, of course. So, however, what we are going to see is more what we see in a bacterium like E. coli, because there are many things happens in eukaryotes, and we don't know exactly their mechanisms. So, assuming you have a double-stranded DNA, and you want to replicate it, what will happen? I will show you a picture. Just I want to use your imagination first. Imagine you have two double-stranded DNA. They want to get replicated. It means replication. It means duplication, double. They have to separate first. Or we use scientific word melt. Separation, it melts. So imagine two strands, they are going to be opened, melt in one region. Not all chromosome will be open, only one region will be open. And then the polymerase enzyme I told you is going to use a single strand DNA. SS, it means single strand DNA, as a template. A template is just the DNA strand which is going to be responsible for the synthesis of the new strand. In prokaryote organisms, this single unique nucleotide sequence site is called origin of replication. So basically, the site where the DNA is opened is called origin of replication. And most of the time, this site is rich in 80 bases. And as we said, the 80 bases are not strong like CG. And it makes sense here. So because 80 is easy to open, so it will be easy to be as a replication, the origin of replication. In eukaryote, though, the replication starts in multiple sites because the chromosome in human are bigger. So if only one site is open, it will be impossible to replicate the whole genome. So if you look at this image, if you look on the A, which is in prokaryotes, and B in eukaryotes, you see in A, you have the origin of replication where the 80, as I mentioned, is there. Then there is a local opening. On the left, you see this is a bacterium. However, in human, for instance, eukaryote, you have many sites opening because the chromosome is big. And this site, when it's open, is called the replication fork. This is the replication fork, and this bidirectional. What does it mean by direction? It means it goes in both sides. One side here is opening and one side is here. And then you have two cells, two daughter cells with two replicated DNA. The same on the right side for a human. The only difference is just you have many origin of replication. So the initiation of DNA replications, when it's open, there are some proteins are involved in this opening. Why it needs proteins? Because if you open the DNA, it can close up again. So you need protein to keep this strand open. And again, when you open a DNA, you open it, start to open it, you might have, you need something to open this DNA. It goes along and open the DNA. So what are these protein? We call them DNA A protein. This will help the double helices not to close. The DNA helicases are the DNA that keep open the DNA, and the single-stranded DNA, this will not let, there are some enzymes that can degrade the DNA. So this single-stranded DNA protect the DNA strand. So the DNA protein, the first one, bind to a specific nucleotide sequence at the origin of replication, causing AT-rich region in the origin of replication to melt. We talked about this. So the DNA protein will come and attach to the AT-rich region, and this start to melt. It means it will start to open the DNA. And therefore you will get a single strand DNA instead of a double strand. Then enzymes called helicases will come and bind to the single strand DNA near to the replication fork, and then moves along to unwind or open the double helixes. And these helicases requires DNA, energy. So if you look, this is to clarify what I'm saying, you have a double-stranded DNA here, okay? The protein, DNA A proteins will come here, and then the helicases will come here and move along to open this DNA, and it needs ATP, energy, it needs energy. And these are the single strand DNA binding protein that keeps the DNA strand separate. So the single stranded DNA binding protein, they are keeping the double strand to be opened, and the helicase, DNA helicase, is moving along to open the double strand. Now there is a problem. If this moves along, there is a problem that the DNA will be stuck, like what we call coils. This is the direction of the helicase. At some point, when the DNA is opened, it will form this, what we call positive supercoils. So, and therefore the DNA cannot move further. Alhamdulillah, there is another enzyme called DNA topoisomerase that will help this to be open in order that the replication can carry on smoothly. So you need to know these enzymes, what usually they do. So the topoisomerase read the DNA synthesis, helicases open the DNA strand, proteins will protect the DNA, and the topomerases will help the DNA strand to be opened. There is a drug called ciprofloxacin is used to treat some infection caused by certain bacteria, and the mechanism of this antibiotic is to not let the topoimerase 2 to do its job, and therefore these positive supercoils are always there, they are not opened, and therefore the DNA replication cannot occur and the bacteria will die. Now, when we look at the direction of the replication fork, this is the direction. There is very, very important concept to know that DNA polymerase the DNA polymerase reads from 3 primes to 5 primes. Please focus with me in here. This is 3 primes, 5 primes direction. The DNA polymerase. So the DNA polymerase, we said the DNA polymerase is the enzyme that helps the replication. It can only read from 3 primes to 5 primes. Look carefully, reads this way, 3 primes, 5 primes, but synthesize from 5 primes to 3 primes. I repeat, because this is a very important concept, that the reading of the DNA polymerase is from 3 primes to 5 primes, but the synthesis is from 5 primes, 3 prime. It means that the new synthesized strand will have a 5 primes, 3 primes. This is one important information you need to know. The DNA polymerase are only able to read the parental nucleotide sequence in the 3-5 direction, and the synthesize, you see, we've written them in red. The new strand in the 5 primes, 3 primes direction, which means the anti-parallel, the opposite. So basically, this is one important information you need to know. So there is another information. We have what we call a leading strand and lagging strand. Lag in English in general means behind. So if you look at this image, focus carefully. I am going to talk about the, to point out to the yellow. This is the 3 primes, and it ends up at the 5 primes. And this is the helicase, DNA helicase. It's open up in this direction. You see, when it reaches from 3 primes to 5 primes, in the direction of the DNA helicase, so the duplication will be continuous. There is no problem at all, because 3 primes, 5 primes, and the synthesis is from 5 primes to 3 primes. This is the leading. The lagging strand in this, the other side, this one, we said that is important that the reading will be from 3 primes to 5 primes. However, in this case, is the opposite of the direction of the fork. So basically, the DNA replication will replicate the strand by segment. So it reads, it duplicates this. Then when it's open on this side, it will replicate one segment. Then when it open another one, it will open. So it will not be continuous. The conclusion is you have 2 types of strand, a leading strand, which is synthesized continuously. The lagging strand will be synthesized discontinuously and by fragment, and these fragments are called Okazaki fragment. So this is what we need to know about this. information. Now, if you want to start to replicate, you have a what we call a primer. The DNA polymerase cannot read, if you have a nucleotide, the DNA polymerase cannot read straight. It needs what we call a primer to start reading. So the DNA polymerase cannot start the synthesis of a complementary strand DNA on a single stranded template. So if you have this is a nucleotide, cytosine, see this is a deoxycytosine triphosphate, the DNA polymerase cannot come and add to it. If this is C, it needs G here. It cannot come and add G. It needs what we call a primer. So this RNA primer will first bind to the DCTs through the hydroxyl 3' hydroxyl group. So this is your DCTP. The DNA polymerase, in order to read it, it needs a RNA primer. So RNA primers will bind here and then form what we call uracil for DCTP here and the primer. So you have the primer, as you can see here, and you have the DCTP. Once this is formed, then the DNA polymerase can come and add to the chain. So the hydroxyl group serves as the first acceptor of a nucleotide by action of DNA polymerase. So basically, the DNA polymerase need a primer and fix it to the, not automatically C. It can use any. It's just as an example here. So it will fix it here and then start to read the whole, adding one by one. What you need to know from this slide, there's what is RNA primer. RNA primer is a piece of RNA that needs to be attached to the single strand nucleotide of a DNA in order to start the replication. So there is another, it's all about terminology. That's why you have to sit down and write down this terminology, and for each of these you need to know what are their function, because most of the question will be coming on what is RNA polymerase, what is DNA polymerase, what is a primer. So the terminology is very important. So there is another one here, what we call a primase. So the RNA polymerase called primase synthesize the short strand of RNA. So this RNA, from where it comes from primase, what we call a primase. So it's responsible for the synthesis of the RNA. In the resulting hybrid duplex the U in RNA pairs with A in DNA. Here, someone might ask a question. You say, oh, doctor, but we know that in DNA, there is no uracil. But here, it might happen because the RNA primer can be uracil. So we say, yes, uracil can be here and can attach at the beginning, but at the end, this uracil will be cut. So at the end product, you will not find uracil in the DNA. At the beginning, yes, you can, but at the end, if the uracil has been incorporated, it will be cut. So there is another one called the primosome. The primosome basically is a complex of the primase that makes the RNA primer and the protein. So this is all you need. What is a primose? You have RNA primase, which synthesizes the RNA primer, and you have a primose, which is the primase plus protein. It's easy just to know what's there, what are they. So basically, how this chain is elongated. Once you open your DNA, once you opened your DNA, there is an elongation. So the DNA polymerase elongate new DNA strand by adding a deoxyribonucleotide one by one at the three end. We talked about this. And depends on what nucleotide are available. So you will build up your sequence. There is another DNA polymerase called DNA polymerase 3. It's the DNA responsible for adding the nucleotide. So very important to know is what adds to this chain, what is the enzyme responsible for adding the nucleotide, deoxynucleotide to build the DNA is called DNA polymerase 3. And in order to make your DNA, you need to have all these deoxyribonucleoside available, the DATT, DLP, DCTP, all we mentioned before. If any one of these nucleotide, deoxyribonucleotide missed, the DNA will not be completed. Okay, so there is one proof reading that the DNA polymerase 3 is very, very accurate and can what we call proofread. It means can read carefully and can make correct any mistakes. So if any mistakes happen in the DNA synthesis, the DNA polymerase 3 can rectify the mistakes. And what we call proofreading activity 3,5 exonuclease. If there is a problem, it can cut and correct